great pleasure to be here, and it's every time I hear about Biobank and what's going on, it's just totally awe-inspiring, and it's fantastic. And thanks to you guys for volunteering. It's just a tremendous resource, and it's really going to move health on forward. So I'm a psychiatrist, so I'm interested in common psychiatric disorder, and I'm interested in depression. And I haven't got a title slide because I'm allowed to be a bit weird. I'm a psychiatrist, okay, and the title is about 3 lines long, and by the time I'd explained it, it would have been done as a talk. So I'm just going to dive in right away and talk about what we do and what we don't know about depression, and what we're hoping that Biobank will help us to find out. I'm very interested in the brain. Well, I'll come on to that in just a sec. I'm not going to talk much about the brain. First thing to say, of course, is that depression is unbelievably common, and we read about this all the time in the newspapers, about how common it is. About 20% of us will develop a depressive illness at some time in your life, our lives, and by that we mean a period of being so depressed that you can't function normally, and it's interfering with things like sleeping and eating and so forth in a normal way. But of course, there's a huge range of variation in that. But it's one word. We speak about depression as if it was one thing. We don't know much about what's going on in the brain, but as I say, I'm not going to speak a lot about that. There is no neurochemical abnormality that we know about in the brain. We do know that antidepressants like Prozac work by increasing serotonin in the brain. You've probably heard of serotonin, and it's a neurotransmitter that's made from tryptophan, which you eat every day in your diet. And that's how they work. They definitely work on that. And nearly all of them work in that way. So we're still on one thing. But depression is so common and so variable that it must involve lots of different mechanisms. And that's why we're not seeing and understanding the mechanisms in the brain, because we're not dividing it up. And that's our whole point, really. We hope that by using Biobank, we'll be able to subgroup depression into different kinds, and it'll overlap with all sorts of other medical illnesses, and we'll find genes and find new treatments in that way. We do know that there must be risk genes because it runs in families, and you can figure out how heritable it is from twin studies, and those studies suggest that about 35% of the risk, so it's not that great, 35% of the risk is due to genes. So they must be out there. And we've been able to show, as Tim said, that we've been able to scan the genome just for the last 15 years. It's amazing how that's moved on. It's just £1,000 a scan now. But we haven't found, using those sorts of studies, a single gene that's definitely involved, that variation in that gene predicts risk of schizophrenia. It's definitely going to be lots of little genes that are involved. And it's similar to the position that we were in with schizophrenia about 5 years ago. So schizophrenia is less common than depression. I'm just going to have a little diversion into that. But it's 70% heritable. And for schizophrenia, we had nothing. And now it's fantastic. In the last 5 years, we've got 100 genes for schizophrenia. And I just want to speak about how that's come about and whether we might be able to do something like that in depression. So I'm delighted that Tim explained about SNPs and things, so that'll save me some time. This is what a scan looks like. So the SNPs are arranged in terms of chromosomes along the bottom there, going from 1 to 22, and the X chromosome is on the far right, as you can see. And each of those dots, well, there's about 1 million of them. You need 1 million to mark bits of DNA throughout the genome. And the reason they're split up is that you're simply asking the question for each one of those SNPs. Is a particular variety of that SNP more common in the people with schizophrenia than it is in the controls? That's what that vertical axis is. You can see that in schizophrenia, all along the genome, there are peaks where some versions of SNPs are more common in the schizophrenia sample than the controls, and we've got 100 of those, so that is fantastic. The key point is that there are so many that each one of them has a very tiny risk. It's less than 1% of the risk. So even with all those positive things, we're only explaining like 30% of the genetic variation, but it's a real advance. And no one of them is going to be critical. But if you look at the pattern, then you can see that particular pathways seem to be, genes for particular processes seem to be affected. And that helps us to identify what processes might be wrong in schizophrenia. So that's schizophrenia, but we don't have that in depression. And why is that? Well, partly because it's only 35% heritable. So to get a sample size sufficiently great would take an enormous number. So the only reason we have this schizophrenia is that we're looking at 40,000 cases and 113,000 controls. And the way that's happened is because people, we thought that we might find genes with 100 versus 100. So all those collections have been pooled in a tremendous international collaboration to produce those sorts of numbers. And the numbers have been going up and up and up all the time. And now we've got over 40,000 versus 100,000. Now the genes are beginning to appear. So to do that for something which is half as heritable, I think isn't going to happen. It's not going to be the answer. And these sorts of numbers, though, we're certainly going to be doing it in the biobank because these are the sorts of numbers in the biobank. As you'll see, we have this sort of number of cases of depression and that sort of number of controls. So the fantastic advantage of the biobank is you can do the whole genome-wide thing in one go. You don't have to have 350 different authors coming together, donating their little connections of DNA. There's enough to detect effects, especially if you take environment into account, and that's what we have to do. So the other things we know are that common sense factors in the environment are risk factors for depression. Bad things happening to you increase the risk of getting depressed, and the key ones are very bad things happening to you in childhood, neglect or abuse, recent bad things happening to you, and not having any social support, so you're not as resilient to bad things happening. And we have to include those, I think, to find interacting genes. So people vary enormously. You see people clinically, I see people who've had the most terrible abuse, and it's understandable they got depressed, and we know other people have terrible experiences and don't get depressed. And those things are modified, we think, by genes. So if we put the two together, the power of biobank and this sort of environmental information, then we begin to find genes, we hope, and then get clues about pathways that may be involved. So we have to include that, environmental factors, I think, to find interacting genes. So all those studies I showed you in schizophrenia, there's no information at all apart from the diagnosis. You don't know if they had life stresses or that sort of thing. In the biobank, you've got all that. It's all there, and I'm going to show you how it is. And there may be all sorts of other environmental factors that we don't know about, we never thought about. Those unknown unknowns that confused American Defense Secretary spoke about. So, and we think that diet may be very important, so we're particularly interested in that. And some medical disorders, like the ones you've heard about, obesity, diabetes, and cardiovascular disease, just worth noting, for example, that if you're depressed and you have a heart attack, your mortality is very substantially increased. There's a big overlap between the two disorders, but we don't understand the nature of that. It could be to do with particular patterns of genes. So let me give you an example of a gene-environment interaction. And this is, I like this picture, it's just happy. And we're a happy bunch of people called New Mood, New Molecules for Mood Disorders. It was a European consortium of 13 centres working on depression. Some of us working on mice. Mice get depressed, apparently. Rats. And and humans. So in Manchester and in Budapest, we collected a pathetically sized sample, 1,300 people in Manchester, 1,300 in Budapest, and recorded everything that we thought was relevant, all those life events and that sort of thing by questionnaire, and looked at particular genes that the mice and the rat people said, we think these are important for stress. And one that we looked at, or a set of them actually, this CREB, BDNF, and TRACK-B pathway. And they all, it's a functional pathway, and these genes make neurons grow and encourage neuronal growth. And we didn't see any difference. If you compare the different versions of these three genes in people with depression and controls, there's nothing to find. There's nothing there. When you plug in environment, then you begin to see significant effects. So if you look on the left-hand side here, you can see along the bottom, we subdivided the sample according to how much childhood adversity they had recorded. And on the vertical axis, you see their risk of depression. And so you can see in general that the more they had experienced childhood adversity, not surprising, the more they reported depression. But that was influenced greatly by which version of BDNF they had. So people with this version were less responsive, if you like, to childhood adversity in terms of getting depression, whereas people with the risk allele showed twice as much. It really amplifies the effect of the adverse effect of childhood adversity. So that gene, variation in that gene, doesn't matter overall for your risk of depression. It does matter if you're in a particular circumstance of having had childhood adversity. And so using that sort of data, You can put it, we put it together to make a sort of diagram of what we thought was going on and tested whether it was significant or not. And so we plugged in the various things that we measured that came out as significant. So there's a genetic effect. There's whether you had depression or not in the questionnaire measures. There's a thing about personality here, rumination, whether you'd had life stress, childhood adversity, and you can make this sort of map. So you see here childhood adversity acting through, being modified by these genes to influence the risk of lifetime depression. And that's what I think we need to do on a biobank scale. That's what we're trying to do exactly that, but do it across the whole genome and all the environmental things that we think of that might be relevant, and plug in diet and other unknown unknowns that could be relevant and could fit into that equation. And we're not going to end up with one map, but end up with several of them. And the genes that we pull out in that way will give us clues as to what's going on in depression and how we might prevent it and put it right. So the practical aspects of it, what have we got in biobank? So first of all, we need to, this is the general stuff from 500,000 subjects. So you can see we have, we know about the age and sex and medical conditions they've had, what medication they're taking, how active people are, whether they've had life events, we've got a record of that, self-rated. We're a bit lacking in early risk factors, the early psychosocial adversity, but there's a new questionnaire going out which is going to address that. There's lots of information about diet, as you'll see. We can use impedance to measure how much of that body fat you've got. We think that might be quite important. And in 172,000 subjects, maybe some of you did the mental health questionnaire, and that tells us about things like depressed mood. So we know that 90,000 of the 500 of that 172,000 felt depressed for a whole week in their life, which is getting on for the level that I mentioned at the beginning. And you can see that. This is the sort of thing you see on the touchscreen. Maybe you've been and had a look at the data, but you can pull this down for every question, and it gives you all the basic information, the mean and the standard deviation and all that kind of thing. And 90,000 felt depressed for a whole week in their life. So we have to decide how we're going to group this. There's a bit more detail here. So the mean length of a depressive episode is 16 weeks. That's quite a long time to feel depressed, actually. It's a lot longer than 2 weeks. And the mean number of depressive episodes was 6. 6 per person who felt depressed had it 6 times as a mean. So it's, there's plenty of variation out there. And so attempts have been made to translate what we know from the biobank questionnaire to make a diagnosis, so we can categorize people into, according to whether or not they had different levels of severity of depression. So you can see a quarter of that segment of 172,000 have depression of one form or another. Very more serious, recurrent major depression, severe, 8,000. Single episode, 7,000. Recurrent major depression, moderate depression, that's when you start treating with an antidepressant perhaps, at 15,000. And the rest don't have it. So those are the sorts of numbers you saw in the Schizophrenia Consortium, all in one place with all sorts of other data to go along with it. So that's how we're going to group it up. That's what we're doing, and we're looking at how to look at influences on that in the environment, from the environment. So we've got to organize these influences on depression, the demographic and psychosocial stuff, diet and body composition, medical disorders, and the genes. Maybe, I don't know if this is the time to say it, but I got an email yesterday saying the genes are ready. They're going to be downloaded from 150,000 people. That's 150,000 scans with those 1 million markers, and the 72 million imputations are there, ready to be used. So that's just happened. So the rest is preparatory work we've been doing towards that. So organizing the demographic and psychosocial data, so we're going to record and put into our equations and models gender, ethnicity, socioeconomic status, recent life events, self-reported functional impairment. That's a key aspect of diagnosis. Can you live your life normally with your symptoms? Childhood trauma we will have in due course. Measures of social support are already there, and measures of life satisfaction. So all of those would be individual items in our big equation trying to predict depression. And this is the data about food, about diet, organizing that. So that takes a lot of organizing. There's a huge amount of data on it. There was a 24-hour recall thing that was done online. So what have you had to eat in the last 24 hours? And your dietary habits. So you can see the sorts of things: cooked vegetable intake, salad, blah, 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 oily fish intake. And so using this sort of information, we can put people into categories that we can then put into our models. For example, there's some evidence, it's not very strong, that the Western diet is more of a risk than the Mediterranean diet. Having lots of cereals and having oily fish is protective against risk of depression. So we can slot that in and see if it's modified by genes and see if it helps you cope with stress or it doesn't help you cope with stress. All sorts of possibilities that we can be evaluated. And so people have been doing work with the database already and extracting from those diaries actual nutrients. So alcohol, calcium intake, carotene, energy, all those things are there. The ones we're interested in are carbohydrate intake, folate in blue, protein, vitamin B12, vitamin D, and you've heard of polyunsaturated fats, PUFAs, and that's the Mediterranean diet. And so sorry, did I say something funny? PUFAs. So we're analysing that. So we're using the data. You can calculate from the dietary history how much polyunsaturated fatty acids you have in your diet, whether it's a lot or a little, and so on. And of course, tryptophan, which I mentioned at the beginning, does have, it's an important factor because it's a precursor of serotonin, which seems to be, a unifying, I suppose, aspect of depression. So we certainly want to know about that. And there's good evidence that variation in tryptophan in the blood influences risk in a small way. And then there's a medical disorder where we think there may be overlaps. It's to give you an idea of the complexity. We're working on this at the moment. And again, just on the other slide, when we calculated those things, we put it back into the biobank so other people can use those PUFA data that we've calculated to do their stuff with. So it's a sort of self-building in a way. So people said what medical disorders they'd had, and there's a list of them, and we put the psychiatric ones at the top. You probably can't read it, but I can't read it. But there's depression in red, and then so there's 526 of those, which is quite a lot to put into an equation. We've got to simplify it and boil it down. So we've recast them into disorder groups like cardiovascular disease, CNS disorders, and at the bottom there, according to the mechanism, inflammation, for example, would be a mechanistic diagnosis. So there are many, many kinds of inflammatory disorders, a huge amount of interest in inflammation and depression. So those boiled down variables will go, and I can show you a bit of data about that. So this is a diagram from the biobank, and what Gabriela Juhatz, who's really the driving force in this study, as you'll see in my title slide, did was to see the comorbidity between these disorders. So there's depression, there's a number of people, the size of the circle says a number of people who said they had depression, and then there's the size of these circles is the number of people who had obesity, hypertensive diseases, allergy, musculoskeletal diseases, and so forth. And the thickness of the line says how much they occurred together. And so we can see what the probability is of that happening by chance and whether there is genuine overlap between risk of depression and these other medical disorders, which might point to common genetic mechanisms, but it might be something like, when you put on weight, you get depressed about it. And so it's, all that has to be teased apart. And that requires elaborate statistics. And so that's, I think, the final point I wanted to speak about, and this is what's happening in Gabriela Juhatz's department. She works half-time here and the rest of the time in Budapest. And they have very sophisticated Bayesian multi-level analyses of relevance to cope with all these predictive variables. Just as a sort of side thought, there's Bayes. He was a vicar in a small English church. 1701 he was born, 1750 something he published a very obscure letter to the Royal Society. He must have had friends to get someone to read his letter, and it was an equation about chance and probability. What's the chance that that your belief is right if you get a new piece of evidence? And that's all I'm going to say about it. It's complicated. Only half Google understand it. And what it does, and so these are all the sort of predictive variables you see in these X's here, and Y in the middle is will be risk of depression, and you can see that basically what Bayes does, instead of looking at all these things individually and whether it predicts risk of depression, it also looks at the interaction between them, and so you can sort of begin to see causal pathways, and including the possibility that depression itself may influence some of these factors that we think are risk factors. So it's a two-way thing. And I hope you understood some of that. I think that's what I need to say about Bayes. So that's the approach. And so to conclude, our aim is to understand and identify subtypes of depression based on interaction between life experiences, personality, diet, medical disorders, and now genes, wonderfully catalogued in 500,000 people in the biobank. And this will help us to identify new genetic and metabolic pathways to depression, enabling us to get away from Prozac and serotonin based on correcting something we know is, we will know is abnormal. And there could well be public health policy implications for prevention, about how we live our lives, the kind of food we eat, and so on. And so I just conclude by saying the Biobank UK is uniquely powerful: sample size, rich description, genetics, organisation, and accessibility of the data to understand and begin to crack the complicated multifactorial disorder that is depression. And here's my title slide. Do you really want me to read that? And there's the collaborators. Gabriella Juhatz works jointly with Semmelweis and the Bayesian types. Jerome Breen works at the MRC unit at the King's College, and they're good at doing whole genome, analysing genome-wide scans, and the Liverpool people know about nutrition. So thank you very much indeed.